Hello, students. I'm here again with a new lecture. This time we will study about eukaryotic cell. Okay. Now, as you all know, the eukaryotic cell, this cell has what the nuclear membrane, the membrane which covers the nucleus, and the doubly membrane bounded organelles are also found there in the eukaryotic cell. For example, we have mitochondria, endoplasmic reticulum, Golgi bodies, chloroplast. So all these doubly membrane bounded organelles are found in the eukaryotic cell. Okay. Now, uh, now we will study about uh, that certain internal organization of the eukaryotic cell is more advanced and elaborate than the prokaryotic cell. So the internal organization of eukaryotic, you have doubly membrane bounded organelles which are found in the prokaryote, which is found in the eukaryotic cell as compared to the prokaryotic cell. So the eukaryotic cells are not identical. Okay. Now we'll study about the generalized structure of the eukaryotic cell. We all know that the eukaryotic cell has cytoplasm, okay, and it has also the doubly membrane bounded organelles in the cytoplasm. Then the eukaryotic cell possesses a variety of locomotory and cytoskeletal structures. What does it possess? It possesses certain locomotory and cytoskeletal structures. All the eukaryotic cells, they are not identical. They differ from each other on the basis of the structure and function. Okay. Now the cell wall is a special membrane which is only present in the case of plant, fungi and some protists. Plant cells also contain a large vacuole and plastids which are absent in the case of the animal cells. While the animal cell, they have the centrioles which are present in the case of the plant cell. So this was about the generalized structure. Now we'll study about the difference between the plant cell and the animal cell. As you're seeing this table here, animal cell has the cell wall, plant cell, animal cell does not have the cell wall, does not have the cell wall. The plant cell, they have the cell wall, it is made up of cellulose. Then the animal cells are round or irregular in shape, plant cells are rectangular and they are fixed shape. The centrioles are present in the animal cells, while in the plant cells only in the lower plant forms the centrioles are present. Centrioles here helps in the cell division. Okay. Now vacuole, it is one or more small vacuole, okay, which is present in the animal cell. Here, while the vacuole in the plant cell is large central vacuole which takes up 90% of the cell volume. Then here the cytoplasm is present both in animal cell and the plant cell. Okay, ribosomes are both present in animal and plant cell. What is the function of ribosome? The function of ribosome is the synthesis of proteins, okay, in a cell. So plastid is absent in animal cell while it is present in plant cells. Plasma membrane, in the animal cell only cell membrane is there, while in the plant cell wall and cell membrane is there. Golgi apparatus is present both in animal cell and plant cell. Okay. Then mitochondria is present both in animal cell and plant cell. Endoplasmic reticulum can be smooth and rough. Smooth endoplasmic reticulum is without any ribosomes, while rough endoplasmic reticulum has ribosomes on its surface. So endoplasmic reticulum is present both in animal cell and the plant cell. Okay. Now chloroplast. Chloroplast, animal cells do not have chloroplast because they do not have the ability to prepare their food. While the plant cells, they have the chloroplast, they have their ability to make food, okay, and they are called as the autotrophs. Autotrophs are the plants. The autotrophs are the plants which can make their own food. Now flagella. Flagella is again the locomotory organelle. Now this flagella may be found in some animal cells, while it may be found in some plant cells also. Okay, microtubules both are present in animal cell and plant cell. Microfilament both are present in plant cell and the animal cell. Okay, cilia is present in the animal cell and it's very rare in the plant cell. Lysosomes occur in cytoplasm in the animal cell. and lysosomes are not found in plant cell. Nucleus is found both in animal cell and plant cell. Cytokinesis in the case of the animal cells. Cytokinesis means division of cell. So cytokinesis in the case of animal cell result in the formation of furrow in the center of the cell. It forms a furrow. While in the plant cell, the cytokinesis is by the formation of cell plate. So this was the comparison of the plant cell and the animal cell. As you are seeing this diagram, this is the animal cell. Here in the animal cell, you have the cell membrane, nucleus is there, nuclear envelope is there, Golgi apparatus, endoplasmic reticulum, okay. Then ribosomes are there, lysosomes, rough endoplasmic reticulum, nucleolus, cytoplasm. All these structures, they are present in the case of the plant cell, okay. In the animal cell, sorry, they are present in the animal. So this is the animal cell. So you have in the animal cell, you are having microvilli, plasma membrane, lysosomes, ribosomes. This was the animal cell. Now this is the plant cell. What is the basic difference? The plant cell has the cell wall, while the animal cell has only the cell membrane. The plant cell has the cell wall, cell membrane. Then it has vacuole, which covers 90% of the plant cell. It has nucleus, nucleolus, nuclear membrane, smooth endoplasmic reticulum, chloroplast, ribosomes, okay. Then mitochondria. All these are present in the plant cell. So this, you have studied the difference between the animal cell and the plant cell, okay. So now we will study the component of the cell membrane. Component of the cell membrane. Now every living cell is covered by a thin, elastic, transparent, semi-membrane, permeable and regenerative membrane. Every cell, it is covered by a very thin, elastic, transparent, semi-permeable and regenerative membrane. This is called as the cell membrane, plasma membrane or the plasma lamella, okay. It is called as the cell membrane, cell membrane, plasma membrane or plasma lamella. In 1950, with the help of the electron microscope, the detailed structure of the plasma membrane was studied on the human RBC. You must be knowing what is the full form of RBC? The full form of RBC is red blood cells. So human RBC was studied in 1950, and with the help of electron microscope, the detailed structure of the plasma membrane of the human RBC was studied. Okay. Now we'll study the structure of the plasma membrane. Now this plasma membrane, it, just see this diagram. This is the plasma membrane diagram, okay. So this is the human RBC, and concluded that this cell membrane consists of what? It consists of lipid, which forms a bilayer with the protein molecules embedded in it, okay. So the plasma membrane is actually composed of a lipid. It is composed of a lipid which forms a bilayer with protein molecules embedded in its places. Later it was revealed that the cell membrane also possesses protein and carbohydrates, okay. Now this lipid molecule, these are the lipid molecules, they are amphipathic in nature. These are what? Amphipathic in nature. What does this mean, amphipathic? Actually, they have two ends. This lipid has two ends. One is polar hydrophilic end. This is the polar hydrophilic, and another non-polar hydrophobic end. Polar hydrophilic end is the region in the form of water-loving head. It is in the region of the water-loving head, okay, which faces towards the outer sides of the cell membrane. This is the water-loving head. It faces towards the outer sides of the cell membrane to interact with the aqueous environment on both the sides. So this is the polar hydrophilic end of the lipid bilayer. Now the non-polar hydrophobic end. This is the non-polar hydrophobic end. Okay, this region is in the form of water repelling tail. Both ends they faces each other that occur toward the center of the cell membrane. Okay, the portion of the lipid molecules. Now the portion of the lipid molecules varies in plasma membrane of different cell type. The lipid molecule portion varies in different plasma membrane. Okay, they are found they are formed of lipid are formed of cholesterol 25 to 32% and phosphoglycerides or phospholipids 55 to 75%. Okay, so this was about the structure of the plasma membrane, generalized view of the plasma membrane. Now you will study the plasma membrane is composed of lipid which forms a bilayer protein molecules embedded in certain places. Cell membrane also possess proteins and carbohydrates. Cell membrane also possess what? Proteins and carbohydrates. Okay, so now this is the membrane proteins. Their membrane proteins are also there. There are two types of proteins in this membrane. That is first one is intrinsic protein and another one is extrinsic protein. Now intrinsic protein, they have stronger association. Intrinsic protein, they have stronger association and they bound firmly to the membrane. As you are seeing, this is the fluid mosaic, this is the integral protein. Okay, this is the lipid bilayer. Okay, and this is the protein, this is the integral protein. So this integral protein has strong association and bound firmly to the membrane. These proteins are buried partially or totally in the phospholipid bilayer. Now peripheral proteins, these are the extrinsic proteins. They have weaker association. They have what? The weaker association. They have weaker association and are bound to the lipids of the membrane by electrostatic attraction. Okay, so this was about the fluid mosaic model. Okay, now these carbohydrates are also present. Carbohydrates are also present. It constitutes about 1 to 5%. So carbohydrates are also present in the cell membrane. Okay, and it constitutes how much? 1 to 5% of the chemical composition. It constitutes how much? 1 to 5% of the chemical composition, carbohydrates, and they are associated with the phospholipids. Okay, they are associated with the phospholipids or with the peripheral proteins to form glycolipids and glycoproteins, etc. So, or they are associated with the phospholipids or they are with the peripheral proteins to form glycolipids and glycoproteins. Okay, now this was about the carbohydrate which is also present in the plasma membrane. Now we'll study the fluid mosaic model. This fluid mosaic model was given by Singer and Nicholson in 1972. According to this model, the lipid bilayer and the integral protein, they appear like a mosaic arrangement, and the quasi-fluid nature of the lipid which enables the lateral movement of protein within the overall bilayer. So this model, fluid mosaic model, was given by whom? Singer and Nicholson in 1972. And according to this model, this lipid bilayer and the integral protein okay, and the integral protein appear like a mosaic arrangement, and the quasi-fluid nature of the lipid which enables the lateral movement of the proteins within the overall bilayer. This ability of proteins to move within the membrane indicates the fluidity of the lipid part. It indicates the fluidity of the lipid part. So this was the fluid mosaic model. You must be seeing a diagram. This is the this is the hydrophobic end, and this hydrophobic end is this one. Okay, and the hydrophilic end is this one. Okay, this is the integral protein. Sugar proteins are attached, lipid bilayer. Okay, so this is the fluid mosaic model of the plasma membrane. So this much is for the lecture now. I hope you have understood this lecture. If you have found this lecture informative, interesting, useful, please subscribe to my channel and like my channel. Okay, thank you.